95, 96 Henderson lecture. We do pause just for a moment to introduce, for those who don't know, and to welcome back to those who do, our good friends, Dr. and Mrs. Joseph Henderson. Joe and Betty, would you stand so that we can say welcome with a round of applause? As you'll see indicated in your program, Dr. Henderson was here as a professor of education, chair of the department, and Mrs. Henderson also spent a good portion of her life in education, helping the teachers that we sent out to teach those children whom we sent to the schools. So it's been a network of education, and Joe and Betty, we are, as a faculty and as an institution, ever so grateful to you for your vision and generosity which has made this lectureship possible. It's really a wonderful contribution to us, and we are forever in your debt for your having done so. Tonight, we also have present here a number of the previous Henderson lecturers, and I would like to acknowledge their presence, ask them to stand, and at the completion of the short list, I would like to suggest we give our colleagues a round of applause. I think Dr. Peter Mackey was here earlier. Peter, would you please stand? Dr. Jim Perkins, Jim. Dr. James Hall, Professor Hall, is here a minute ago. There he is. Dr. Sharkey, Gene Sharkey, and Dr. Alan Gillis. Those are previous lecturers who are here. We welcome you. It is not really necessary to say anything in addition to the biographical data about Dr. Becker, but one is inclined to say something anyway. And that is that in addition to all that is on here about Dr. Becker, what is not stated is her example as a teacher, scholar, and researcher. What is not on here are her many contributions to our campus life. I can't think of many committees that I'm aware of where over the years Monica hasn't been a servant on some of those committees. She has distinguished herself as an associate trustee. She has served search committees of this college, and the list goes on and on. In addition, it's a joy after, for example, myself, for me, having been here into my 9th year, where people are no longer just a professor, but a real human being. You get to know people because of their wonderful qualities. And so, Monica, let me say to you, I'm especially proud to introduce you for this Henderson lecture, not only as a distinguished professor here at Westminster College, but as a cherished friend. Dr. Becker.
I'd like to also thank Joseph and Elizabeth Henderson for providing me the opportunity to give this speech tonight. It was very good for me to be reflective about the kinds of things that I've been doing and what I would like to do in the future. I also want to thank you all for coming, because you get a little bit apprehensive that nobody will come to the talk. So my first apprehension is satisfied. There are people here who are willing to listen, so thank you for coming. Especially to the freshmen who have a test in 2 days. So I know who you are, okay? The title of my talk, From Gene Jockey to Photo Finder, sort of reflects what I've been interested in, but actually John Deegan suggested it would have been better title to have Gene Jockey to Photo Finisher. And I like that a lot better, so I'm changing the title right to start with. It's going to be From Gene Jockey to Photo Finisher, thanks to John Deegan. Now, when I started here in 1985, I knew that what I wanted to do was to do research with undergraduate students. I think it's essential that students in sciences actually do science in order to learn it. It was something that I didn't do that much when I was an undergraduate. And it gives you a whole different perspective of what science is. So I set out to do science research at Westminster College, and I was open to almost anything. And I did almost anything. I was the electron microscopist. I had one student who worked on eye development because she wanted to be an optometrist. I had another who worked on teeth because she was going to dental school. Another student who wanted to work with corn seedlings. So in 1 week, I'm trying to figure out how to grow corn in boxes, how to brush the teeth of a gerbil. Do you anesthetize it or don't you? What do you do? And those were good projects, and I think they were good for the students, but it was just too broad. It didn't allow me to really focus or to get proficient in any one area, so that I would like to have had one student continue a project from one to the other so that we could build on that information. I also wanted to get to know people in the field so that I could help them get into graduate schools and that sort of thing. So when my sabbatical came up, one of the things that I felt I had to do was focus on my research. I had to choose an area that I could become very proficient in, and then I would have my students work on that area. That was my first goal in my sabbatical. My second goal was that I wanted to learn some techniques in molecular biology. We had just started a molecular biology major here on campus with Dr. Patrick McCarthy, and there were a whole lot of techniques that are very difficult that we weren't even thinking about when I was in school. And it was only if you were in school at that time in the 80s that anybody really was proficient in those areas. So I wanted to become a gene jockey. That's what they call themselves. I was lucky enough to get a sabbatical at DuPont. Some of you have heard this, but it wasn't due to my terrific accomplishments. It was because, see, I shouldn't even say this. I should just let you think it was due to my terrific accomplishments, right? It was because the person who's head of all the sciences there happened to be a colleague of mine when I was in graduate school. And he remembered me favorably. So I managed to get this position at DuPont with really top-notch scientists. And I tell you the truth, the first week I just walked around thinking, oh my God, I'm here, I'm here. That's it, I'm here, I don't have to worry about anything else, and I'll just take one day at a time. And I was so uptight for a week that finally someone stopped me and said, please, please, Monica, just make a mistake already and get it over with. We'll all feel better. All right, well, the thing that I was supposed to do, before I get into that, I want to tell you a little bit, sort of a background, because many of you may not really know what molecular biology implies. So, this is a DNA molecule. How do I do this here? Oh, we do this like this. This is a DNA molecule right here. It's double-stranded. We could work this. It's double-stranded. You could think of this as being one area that's a gene. Now, when a particular gene is going to be activated, the double-stranded opens up. The double-stranded molecule opens up, and there is a RNA molecule that looks very much like the DNA molecule. Now, notice that you have here regions that keep the two molecules together. And those are nitrogenous bases that match up. So the A always matches with the T, and the G always matches with the C, right here. These open up. And then when this RNA molecule is formed, again, you have A's matching up with T's and G's with C's. Not completely true. RNA has uracil instead of thymine, but otherwise they're very much the same. So you get this messenger RNA molecule, which is going to be made into a protein. And it's made into a protein because 3 nitrogenous bases will code for a different amino acid. There are 20 different amino acids that make up all proteins. Now look around you. All these people you see, mainly what you see is protein. The hair is protein. I'm looking at Oscar. The hair is protein. He has hair. The skin is protein. The muscle is protein. Most of what you see is protein. Now there are some things in our bodies that aren't, like bone and fat, but that's made by protein. So everything that gets done and when it gets done is determined by the protein that is made in the cell. And all of that protein is coded for on the DNA. Now, what happened in molecular biology is that we figured out a way to break into this DNA. We figured out the code, We figured out a way to cut the DNA. We could recombine the DNA. And what scientists found out in the 80s was that you could recombine this DNA from different organisms. You could take bacterial DNA and put it with human DNA. And you could put it into a bacteria and it would code for that protein. Now this was pretty revolutionary because it meant that we could just recombine all kinds of things. We could pull out genes, we could do everything the cell did. So all kinds of new mechanisms were developed in order to study the genes. Before we always had to study the proteins. What does the protein do? But now we could just study the gene. Put the gene into something else, make the protein, change the gene, see what happens to the protein. So this was the revolution in molecular biology. Now, what I was to do at DuPont was I was to isolate a gene. I was to isolate a gene that was called a binding protein, this blue thing right here. All right, now think of this little engine as the starting of this RNA molecule. This engine's got to sit on the DNA and chug, chug, chug along to make the RNA strand. But it can't get good footing. This is where it sits in the red portion. It can't get good footing. So in order to hold that engine down, to really get it started, you need a DNA binding protein that holds it in place and gets it started right there. So actually it's this DNA binding protein that determines that these genes are turned on. Now the particular genes I was interested in were the ones that made all the elements, all the proteins that you needed for photosynthesis in photosynthetic bacteria. These are bacteria that can photosynthesize under anaerobic conditions. So when there's oxygen there, they stop photosynthesizing and they start just using the nutrients in the media. As soon as there's no oxygen, they start photosynthesizing. Now you would think they would depend a little bit on light, wouldn't you? Freshmen, wouldn't you think that if you're going to photosynthesize, that's what their test is on in 2 days, that they would use light? Well, they don't. They use oxygen. And so what this lab wanted to find out was how were these genes being turned on and off? And a good way to determine that was to find out more about this binding protein. When is it produced? How is it produced? What is it really? Does it react somewhere else in the DNA? So that's what I was supposed to do. I was supposed to find the gene for this binding protein. As most of this kind of work, if I were to find the gene and the protein and everything, it would take about 5 years. I only had 1 year. And most postdoctoral positions are for 1 or 2 years. So they are really kind of collaborative. One person finds something, the next person adds on to it. So the young man who was there before me, had isolated a lot of sections of DNA, and he had isolated a lot of this DNA binding protein. Now, if you get enough of it, you could put it in, and the machine looks nothing like this, but I thought this looked very scientific. You could put a lot of this isolated protein into a machine, and it will tell you what the first 15 to 20 amino acids are in that particular protein. It's called an amino acid analyzer. And of course, you know, they're very expensive and only certain people know how to run them, and you trust your protein to this person and hope they put the right one in to give you your sequence. So what you get is you get, say, the first 15 to 20 amino acids. Now, you could figure back what the DNA would be for that little section of that gene because I told you that 3 nucleotides code for a particular amino acid. It's a little more complicated than that, but let's just leave it there for now. You could determine what the code is for those amino acids. Are you still all with me? Okay. So what you could make is one little end of that gene. You've essentially figured out one little end. It's only 16 to 20 amino acids, and most of these proteins actually have like 1,000 amino acids. But it's enough to give you a uniqueness for that particular gene. Now what you do is, again, you get some lab to make, you tell them what your sequence is, and they make this DNA, this little strand of DNA, and label it with radioactivity. Now I'm telling you this because this is like a general procedure to pull out genes. It's not something that I just did, it's general procedure. It's called Southern blotting, and it's really named after a man named Southern, so it's not because it has anything to do with directionality. And you take the DNA and you cut it with particular enzymes that cut the DNA in certain places. And you could run the DNA. Now this is like if I took your DNA and we're going to find this gene in your DNA. We take his cells, spin them down, isolate the DNA, cut it. This restriction enzyme always cuts it the same way. Run it on an agarose gel, which is like gelatin, a gelatinous kind of thing. And you run it, and they run different sizes. And then you put that piece of DNA that you made that has a label on it, you stick it in there, and you've made this single-stranded, and you allow it to line up. It's going to line up where the nucleotides would match up with what it is, right? So you get, remember, each of these nucleotides finds one that it matches up with. And you could find the portion on the nylon filter which has that particular section of the gene. Forgot to say here that you have to put the gel on a nylon filter. And you do this by just smashing it like capillary action, and it comes up just like a blotter. All right. All right, once you find the section, you could go back and cut out, you run another gel, cut out that section, and now you've got the piece of DNA that has the gene that you're looking for. You take that piece of DNA and you put it into a plasmid. Now a plasmid is just a little round section that is able to make lots of copies of itself in a bacteria. Don't go to sleep, it'll get better. All right, so here you're growing up the plasmid. You're going to isolate the plasmid, sequence it, and then once you have the sequence, you think you have your gene. Now, how do you know it's the gene you really want? Right, well, after you get the 1,000 nucleotides, you sit at your computer. At that time, all the genes that were sequenced in the world were put into a gene bank. You couldn't publish a paper unless you put your gene into a gene bank. So you typed it into this gene bank. There was a software program. You essentially asked the program, this gene that I isolated looks like this. Does it look like anything else in the world? And everything that's been sequenced in the world, there's going to be comparisons made of what your gene looks like. Now, I did this, and I thought I had a DNA binding protein. So I came back the next morning, and I expected the first 10 things that my gene should look like, because it prints this out for you, should be like other DNA binding proteins. Instead, the first 10 things were indoglycerol phosphate synthase. Well, that's not what I wanted. I wanted a binding protein. And what this gene was a gene that helps make tryptophan, which is an amino acid. Well, I had isolated the wrong gene. Luckily, it wasn't a gene anybody else had ever isolated. There were people that were working with tryptophan that were very excited that I isolated this gene. There were people that were doing mapping that were very excited that this gene was mapped because it was in a whole different part of the chromosome. But nobody in my lab could care two hoots about the gene that I actually isolated. Well, I continued, because I didn't have that much time to start over, I continued and did knockout mutations to prove that this gene really does make tryptophan in this organism, and I was able to publish that, but it didn't make me a hero in the photosynthesis world. But this could happen a lot, and I didn't really know for 3 years later whether or not I had made the mistake or whether the person ahead of me had pulled out the wrong protein. And if someone pulls out the wrong protein and you're using that as your probe, there is no way you could get the right gene. And in fact, that's what had happened. They had just pulled out the wrong protein. You could imagine someone doing their doctoral dissertation on something like this, working 3 or 4 years and then pulling out a gene that somebody else had already isolated. So I was lucky in a way. But when I got back to Westminster, I was going to start a collaborative research lab with 3 students. I had 3 very good honor students, and we were going to all work on the same thing, and we were going to solve this problem. And the problem that I chose was we were going to look at photosynthetic bacteria and determine whether or not what you grew them in, what nutrient you grew them in, made any difference as to how they turned on their photosynthetic apparatus. So if you gave them a lot of nutrients, would they just opt not to turn on the photosynthetic apparatus and just use the nutrients? Because after all, it takes a lot of energy to make all of this photosynthetic apparatus, and they wouldn't need it. They could just get it from, just pull it right in from what they're growing in. So I figured these organisms were pretty smart, and if you gave them enough good stuff, they wouldn't bother doing all this. So that was what we were trying to work with. And what we had was, this is another real, used often in molecular biology, we had what we call a reporter gene. Now here's the normal gene with the little chug-a-lug going along. What you do is you leave the place where the engine would attach, you leave the place where the binding protein attaches, but you put something else back here. That's called a reporter gene. And then here's the bacteria. It's got a normal copy of the gene, so that whatever it has to make, say to photosynthesize, it could make, but it's also got this reporter gene. So every time this gene turns on, this one turns on. So that you could measure exactly when you're turning on your gene and to what extent. We measured it by measuring this beta-galactosidase, but a very common reporter gene right now is luciferase, and that's the stuff that glows in a firefly. So they actually put this in the back of other genes, so you could have tobacco plants that glow. Whenever you have a gene that turns on, the tobacco plants glow. Or the organism, like you could put it in a Drosophila, as it's developing, different parts will start to glow because you have that particular gene being turned on. So that's a reporter gene, and I'll use that concept again. This idea did not work. See, I'm giving you the good and the bad. So far, I guess I didn't pull out the right gene, and this concept didn't work. But I kept trying, see. It didn't work for 3 reasons. First of all, this project required a lot of growth curves. And growth curves are very boring. Students have to measure every 2 hours how they grow. It just doesn't seem like real science, and they hate it. The second reason that it didn't work was that the way we were measuring the enzyme, we had to break open the cells. And we really needed a sonicator. And we didn't have a sonicator at the time. So I devised some other way to break open the cells, but it wasn't consistent enough. It didn't do it accurately enough each time. So the values fluctuated. And the third reason it didn't work was totally surprising. The 3 students really wanted to do their own projects. I found that collaboration at this stage, when they're first starting their project, may not be such a good idea because they have to get the confidence to work on their own problems, and they have to feel like they have ownership. So that did not work. One of the students continued this project, and that was Steve Lang, and he got fairly good results. The other student decided she wanted to work on some PCR. So here's the PCR concept. She decided to look for a particular gene that was found in bacteria and found in humans, and it was almost identical. It was 60% identical. So that's a great span for a gene to be conserved. And she was going to look for it in some other organisms in between. So she chose the mustard seed, Arabidopsis, and she was going to look for it there. But part of the purpose of her whole project was to go through this process of pulling out the gene with PCR and figuring out that it was the same one. Now here is how PCR works. Now some of you might have heard about PCR if you watched the Simpson trial, because even the next thing I'll explain is fingerprinting. You use PCR. It's a machine that essentially heats up the DNA. You've got all the things in a little test tube that you need to make more DNA. So you've got enzyme in there, you've got nucleotides in there, everything you need to make the DNA. You heat it to a high temperature, the two strands come apart. You have to know enough about the gene to know what the starting and the end is. So here's the starting and the end. Those are called primers. You put the little primers in there, and they'll start copying the DNA. Remember when I showed you the RNA? That's okay. Remember when I showed you the RNA, the gene opens up and only one strand is copied. When you're making DNA, the two DNA strands open up and both strands are copied. But they're copied in opposite directions like this. So as long as you know enough about what you're looking for, that you know what the starting and the end is. If you've pulled out a gene in one organism, you should be able to pull it out if it's the same gene in any organism. And this is the method you use. So you make 2 extra copies, then you melt that apart, you make 2 more, and you keep doing that so that the growth is exponential. And after each cycle lasts about 7 minutes, and after about 3 hours, you could have 1,073,741,824 copies of that gene. So it goes very quickly. So now you could pull out genes very quickly. Now this works if you know enough about the gene. You found it in one organism, sequenced it with this southern and everything that I did so you know what it looks like. You know the 2 ends, and now you could pull it out of anywhere. So this was Leslie Bober. She did this project, and she pulled out this gene from the mustard plant, and she found that it was there. The other student was Megan McCarthy, and she was my first student who hated bacteria. I didn't know this when she first started to work with me, but she wanted to do something with horses. She's right now in vet school, and she just loved horses. So her father and I worked out a project that she did, and this was DNA fingerprinting in horses. She would get the DNA, she would cut it with restriction enzymes, run it on the gel. Molecular biology is a lot of boring gel work. If you go to these conferences, they say, just one more picture of a gel. No one wants to see pictures of gels. You blot it onto nylon, and now the tricky thing is you have a little probe. Remember when I made that little probe, I made it to a particular part of the gene? Well, here you're making a probe to a part of the DNA that is variable. It's different in different people, so that you get different banding patterns. You can also amplify one gene and look at that too. There's different ways this DNA fingerprinting can be done. This is what they did in the Simpson trial. I mean, they took his DNA and they probed it, and they found whether or not the sequences were accurate with, used the probe, and they found his pattern. And then his pattern was unique from the patterns of the victims. Well, you could do the same thing with the horses. All right? And she did a very good job on this project. It was a good pilot run, and I think there are actually now, Dr. McCarthy's working probably with 5 students who are continuing this kind of work with horses and doing DNA fingerprinting. Now, as I said, Megan was my first one who didn't like bacteria. But that summer, too, I went to the Gordon Conference, which was, it was an honor to go there. Best scientists in the world go. It was on photosynthesis. They decided to pick one teacher. NIH forced the Gordon Conferences to pick one teacher for every meeting because NIH realized that teachers have more impact on who the next generation of scientists are going to be. And they wanted some teachers involved in this. And this is how I got to go to the Gordon Conference. And it was on photosynthesis. But for me, it was not a great experience because what I saw there was tremendous tunnel vision. Everybody was working on the same 3 organisms. Everyone was working on the same system. And they were all racing to figure this thing out. So I've got some overheads here that just kind of, if you think about a leaf, now this was on photosynthesis, so it could have included a whole lot of things at a meeting like this. If you think about a leaf and a cross section, all right, here are the individual cells in the leaf. Here are the individual chloroplasts in the leaf. Here's a chloroplast with a membrane. All right. Whoops. Let me get this off of here. Here you see another picture of the membrane. All right. What they were all talking about, now how many layers in are we? What they're all talking about was a glob that was in the membrane. And this was the glob. And they weren't even concerned about the whole glob because they'd already worked all of this out and people won the Nobel Prize for it. All they were interested in was this part of the glob, how it was splitting the water molecule. So how many manganeses were there? How many waters were there? And I decided at the Gordon Conference that I was going to switch organisms, that I couldn't work with this organism that everybody else was working with, that I had to get something that was much more remote. So I decided to work with aerobic photosynthetic bacteria, which require oxygen. And they make the bacteria chlorophyll, but they don't seem to use it, which is why nobody was working on it, because they seemed to be a fluke. They made this whole big apparatus, and they didn't seem to use it. And people were trying to figure out, well, why do they make it? Is it just a mistake? Is it that they're evolutionarily evolving to something else? As far as I could figure, there were 3 labs that were working on it intensely in Japan, Russia, and Germany. And the interesting thing that I found about it was that they seemed to determine that some of these aerobic photosynthetic bacteria had a day-night cycle. They made this bacterial chlorophyll, the whole photosynthetic apparatus. They seemed to make it at night and maybe utilize it during the day, so that they were on a circadian kind of rhythm. So this intrigued me, and I thought we were going to start working with aerobic photosynthetic bacteria and determine why they make their photosynthetic apparatus. Well, I had a student at the time, Eric Martz, who determined some of the conditions that this bacteria would grow under. He also determined when it would make the most amount of bacterial chlorophyll. And I was able to get some electron microscopy pictures of it that nobody had ever seen before, and I presented those at a meeting. And last year, Liz Horn worked on trying to determine whether or not these bacteria really did produce more bacterial chlorophyll when they were grown on a day-night cycle, on a sort of a circadian rhythm. And she got some indications that it actually does. This year, I have Lori Amma working on a continuation of this project. So this is my first student that's actually continuing something. Now, here's what I'm looking for. These are pictures that we actually took of our Rhodobacter capsulatus, which was the first organism. You see all the little vesicles that it makes? That's what we're looking for. We found indications of it in this Erythrobacter, but we have not optimized the conditions. So that's what she's working on this year, is to see whether or not by changing the salt concentration or by growing them under different conditions, we could optimize and actually show how these vesicles are formed. And give proof that the vesicles are formed. Because if they don't make vesicles, then they'd have to do this in their membrane structure. Then we could look at the membrane structure. Well, I didn't tell you too much of this because in the last year, what's really happened is, and you're all getting a little sleepy-eyed, people are not all that interested in photosynthetic bacteria. They really don't care. You know, they just, so what? So these guys make some bacterial chlorophyll and they don't know why they make it. I'm not interested in working with that. Students like to work on something that has relevance to them. And I decided last year what I really have to do is I have to expand the research interest in order to get students really interested in doing research again. With me, anyway. Now, what I decided to do was go into photobiology. And photobiology is how biological systems react to photons. It's a much larger area than just photosynthesis. And since I'm not all that familiar with this area, I'm in the process of doing this, of writing a handbook on what kinds of experiments have been done, on what kinds of experiments can be done here at Westminster College. And I plan to include the work of students that have already done work on photosynthesis, so that a student who comes along and wants to do an honors project has an idea of what this field entails. I'm hoping that the other faculty in biology would also have some use for this handbook and would maybe even contribute to it, so that we could have students working on an area where they're still doing their own research, their own independent research, but when they're finished, they could kind of collaborate because it's all connected. So here are some of the areas that have been very interesting. This is what I've gotten. I'm working on the handbook now, so these are some of the things I've completed. The first thing is this DayNight Cycle that I saw in the photosynthetic bacteria. I found that there was an awful lot of information on this. They have isolated cyanobacteria where they have, this is a kind of bacteria, where they have mutants. You change one gene and it changes the NightDay cycle, which usually is 24 hours. It changes it from 16 to 70 hours. So they stopped paying attention to, here's how this works. Now in these organisms here, they've actually isolated one gene that controls this in Drosophila, in all of these organisms. Now here's how one of these clock mutants work. Normally, you have 12 hours, this is night, this is day. All right, so something is active at night, not active during the day. This could be you sleeping. Sleep at night, don't sleep during the day. In Drosophila, it's activity. They crawl around or they try to mate. Well, you might ask, what is it in a bacteria? In a bacteria, what they used was a reporter gene, and they put it behind a photosynthesis gene, one of these glow things, and the bacteria just glows. So it'll glow for 12 hours and then shut off. All right? And then if you put these organisms in all darkness, they still have the same cycle. See, this is how you know you're working with a cycle gene. It cycles night and day, put them in all darkness, and it still cycles. Now, when you're looking at some of these mutants, what happens when you put them in all dark? They might become skewed. Many of them, then, if you put them in all dark conditions, the mutant, you totally lose it. They totally lose that synchronized rhythm. Or they might go to 17 hours, or they might go to 70 hours. So you've got one gene that's determining the night-day cycle. And as long as there is night and day, if you're exposed to night and day, everything's normal. But that thing doesn't keep working afterward. So there's one gene that controls that. And a lot of these genes are very much the same. They found one in mice, and if there's one in mice, there's probably one in men. So there are in humans, I shouldn't just say men. So they're looking for it in humans. They're looking for these kinds of cycle genes. Another area that is of great interest is the area with melatonin. Now, melatonin is a substance that's produced in the pineal gland in the brain. And the pineal gland in our brain is in the middle of our brain, but in many animals it's very close to the outside of the head, and it's really called like a third eye. So this pineal gland is sensitive to light. In our case, it's the eye that reacts with the pineal gland and tells us that there's light. And this melatonin is a substance. You see, these things are produced in the light. So this melatonin is something that is produced when there's no light, in darkness. So once it gets dark, this serotonin is converted to melatonin. All right? And the effects of melatonin, you may have read about it in Newsweek lately. They attribute all kinds of things to this substance. It's supposed to help you sleep. Well, that makes sense, because if it gets dark and you produce more of it, you get kind of mellow and you go to sleep. So people take it as a sleeping aid. It's also used in some animals as triggering fertility, because the length of the day and the night triggers what time of year it is, and so you could have fertility. So they're testing it in places as an anti-fertility drug. Women are taking it as a birth control method. It's supposed to increase your immune system, so they're using it with patients with AIDS. This is the kinds of tests that are going on now. It is also, since at the age of 10, you have about twice as high levels as you do at the age of 45. It was suspected that it's an anti-aging product. And the way they checked this was they took the pineal gland from a young rat and put it into an old rat and reversed the two. And the old rat acted like a young one, and the young acted like an old. I'm not sure. They didn't say what it is that they do when they act young or old. But I imagine it's just more activity or mating or something. So now this is also thought to be an anti-aging device. Now, last year, and one more thing. You see this enzyme right here that converts this step to that step. This enzyme is identical to an enzyme in bacteria that is utilized in the photosynthetic apparatus. It makes a carotenoid. So I was already interested in this enzyme because it was utilized in photosynthesis, and it's utilized in something as complex as making this chemical in the brain. And then last year there was a guest lecturer, Andrea Tilden, who happens to be Oscar's niece. She did her research on melatonin, and she looked for different levels of melatonin in animals, and she has a good assay for this. So I asked if they had ever found it in bacteria, and she said nobody, as far as we know, nobody's ever checked. So we are checking for that. Lori got the samples all ready, and we managed to get them out yesterday to Andrea Tilden, who's going to check to see if melatonin is made in bacteria. I can't really think of any reason why it should be, but it's worth checking something like that because the enzyme is so similar. The enzyme that's doing the conversion is so similar. So it's possible that it's also made in bacteria. But this kind of thing, studying something like melatonin or pooling out genes that are similar in different organisms is much more attractive, I think, to students because they can relate to it more to themselves. As maybe you can too, and everything I told you about bacteria, it didn't look nearly as interested as if I'm telling you about this. A whole nother field that has to do with photobiology is the visual system. And in the visual system, again, there are substances that are identical in bacteria and in humans. Here's an example, rhodopsin. Rhodopsin is used in very primitive bacteria as an energy source. They can get the light photons and convert that directly to energy. Not photosynthesis, but something like it. Okay? There is this gene called the PAX6 gene, which has been pulled out of mice and Drosophila. And this is a gene that's responsible for eye development. So if you take the gene from mice that does the eye development, and you stick it into a Drosophila egg, and now this Drosophila egg has got a mouse gene that would tell the mouse to make eyes, right? What happens in the Drosophila? Well, the Drosophila makes lots of eyes. This one fly had 22 eyes. Here's the front of it. So here's the normal eye, and then here's the extra eye. Well, that doesn't sound too fantastic because the extra eye is next to the other eye. It could have just sort of made an extra eye. But it made an eye on the knee. So there's an eye on the knee. Now, does this fly see with this eye on its knee? I don't know. I don't know if they know, but it had 22 eyes spread all over its body. It's fantastic because what this gene was, this wasn't a gene that makes eyes. This is a gene that tells other genes, let's make eyes. Hey guys, we're going to make eyes. And this eye in a fly is much different than an eye in a mouse. This is a compound eye. We have a whole different eye system. But you see the genes that actually turn on the eyes are still conserved. They're still the same. So you could look for these kinds of genes in a lot of different organisms. And it's easier. It's much easier to do this now because remember I told you when I sat there and typed in my whole 1,000 sequence of genes? That used to be a private enterprise that you had to be connected to. Somebody in Wisconsin, if you paid them a lot of money, they kept track of all the genes and you got to do this. NIH does this now and it's on the World Wide Web. You could pull out any gene that you're interested in. Students, you could do this for any gene that you're interested in on the World Wide Web if you go into NIH. And they even have a software program where you could do comparisons. So we could do this kind of thing now without paying extra money to have this kind of software. So, you know, this is changing how, it's changing the accessibility for this kind of information. Now, there are some other areas too that I plan to cover. Some of these, photo movement. A lot of organisms move away or toward light. Photoimmunology. Again, this has to do mainly with ultraviolet light. Ultraviolet light seems to inactivate the immune system. And you could do all kinds of experiments with ultraviolet light because it damages the DNA, so you get all kinds of mutation rates. And with the ozone layer becoming so much thicker, we're all being exposed to ultraviolet light in greater amounts. So the kinds of experiments that would be involved here would have a direct application. And I think there are some of these things that we could do at Westminster. I'm not sure that we could have too much application here, but I would include them because photochemotherapy is using different chemicals or substances in the body that are inactive until you shine light on them. And then if you shine light on them, they become active. So that you could target where you're going to activate this molecule. Anyway, I am in the process of preparing this, and I am changing my emphasis. I would like to expand it. I would like to include especially a lot of this gene work, pulling them out, finding the genes then with PCR in different organisms, and it really doesn't matter. You could find them anywhere. Just get the DNA and look for them. Eventually, we would like to get a non-radioactive sequencing technique where we could sequence the genes that we pull out. So when you think about this, you know, when you think about how we're finding all of these DNA sequences, there's a project on to sequence all of the DNA in humans, all of the DNA in all living things. So we will know what the genetic code is of all living things. We will be able to do comparisons with computers. Is it the same in the fly as it is in the man? How does it turn it on there? We're going to have this vast amount of information. And if you think like, I think like I have for a long time, when you're at a molecular level, you have kind of a reductionist view. You think, well, once we know all this, we'll be able to control all this. But in fact, that's not true. And that came to me this summer as I was sitting out on my deck contemplating all this. Wow, once we have all these genes, we're going to be able to control all these things. And in fact, we're not going to be able to control anything because we're going to be able to change things quicker. we're going to maybe be able to speed up evolution, but we can't really change something as complex as life because we have no idea what changing one thing will do to many other things. We can't make predictions about that. So when you think about something like living systems, they are not, you can't think about them in a real simple way. You can't say, well, if we have all these parts and put these parts all together, we'll have the answer. And I thought it was kind of a novel idea, but in fact, it wasn't. There were a lot of people that have already thought about this before me. Probably some of them were philosophers. But it's called the science of complexity. And there's actually a whole institute in Santa Fe, New Mexico, where people just study complexities like life, like evolution. And it's hard to explain what we mean by a complex system. You kind of know. You know life is complex. You know evolution is complex. The weather is complex. But these are kind of characteristics that I've put together that would define a complex system like life. It's very unpredictable. You can't make predictions. It has a level of self-organization, such as pacemakers in the heart, or fireflies or crickets. The fireflies all glow together. You put them together and they start glowing together. They may be self-organized, but they have a criticality. And the way that these scientists explain this is with sand piles. If you've got a sand pile, you keep adding sand to it, you know you're going to have an avalanche or you're going to have just a little trickling. You can't really predict which you're going to have. But even if you have an avalanche, you'll still end up with a sand pile. So it's like in a critical phase, you don't know what it's going to do. It's unpredictable, but you know it has a certain amount of order because it's still going to stay the same when you're done. Systems like this can, even though they have an order, they can get very chaotic. You could have something happen environmentally, say to life, and life will go off in a whole different direction. So it's very sensitive to external forces. It's also very sensitive to initial conditions. I have that someplace here. And all of these, if you look at them mathematically, they're very nonlinear. They don't allow themselves, and I have a lot of mathematicians here, but they don't allow themselves to be defined in a linear kind of equation. You increase this, so that you could always tell where you're going. And the kinds of questions that they're asking with this kind of complexity is they're asking, for example, why have living things always evolved to be more and more complex? Now, I spent a good deal of my research life studying bacteria, which were thought to be not very complex. In fact, the cell is very complex. But as you look through evolution, things in time became even more complex. They interacted with each other. Why are things evolving to become more complex rather than less complex? With these kinds of studies, can you explain the social behavior of animals such as bees and ants, which sort of as a group act as an individual? They're a complex system of their own. How do they, as a group, act as like an individual? And I guess this kind of, at what point does the whole become more than the sum of the parts? Because that's what all these complex systems, or at least life or life in a cell, if you put all the parts together, it's more than just taking them apart. If you smash something under a microscope, you can't put it back together. It sort of has to crystallize. Everything has to be working at the same time. And what these scientists are looking for is they're looking for universal laws. They're looking for whether or not these complex systems have some kind of laws that can be pulled out that are the same for everything. Same for the economy, the same for life, any of these complex systems. Some of them think they could find laws like this. Now, I know that I've gone, from the bacteria to photo finisher, to this complexity, which means maybe I've come apart a little bit too much. Started to focus at first and now come out, but this is really intriguing. And I think a study like this, if you combine it with biology in a liberal arts education like we have here, that is integrative anyway, has a lot of potential with different areas of mathematics, different areas of science, even at the economy. So I would like to introduce something like this at Westminster. And this I wasn't even planning on talking about, but sort of came to me this summer when I was actually working on the handbook and thinking about how, what all this means. So I guess I've gone from trying to be a gene jockey to photo finder, and now I think I'm on the edge of chaos. But it's not a bad place to be. So thank you for listening. I'll be glad to answer any questions. I want to acknowledge the students. I forgot to do this. These are the students that I mentioned that had done projects. Actually, I didn't mention Brian, but he is one of the students that said he hates bacteria. He would never work with bacteria. So he was one of the students that encouraged me to maybe broaden my research a little. So he actually is working with me, and we're working on the prion gene. And this is, we're pulling it out using PCR, using molecular biological techniques, but working with something that has a little bit more relevance to him, and we're trying to pull it out of organisms like bacteria. The prion is a protein that is a degenerative disease in the brain. And if you put a prion with other proteins that are normal, they make them degenerative also. So there doesn't seem to be DNA involved here, it's just the protein that affects other proteins. So anyway, we're looking for this in very primitive organisms. So I just wanted to mention him. And these are the people that helped me at DuPont, Deborah Young, and Barry Mars. So, do you have any questions? I hope I didn't bore you too much. Do you have any questions on anything? Yeah.
When you speak of student research, how long do students spend? Do they begin their junior year, fall junior year, and work for 2 years, or?
Most of the students I've worked with work about a year and a half. About a year and a half. Some have worked 2 years, and some only go a year. So they start in their senior year and go through. I've had some independent studies that go for just 1 term or 2 terms. Getting them really interested in something is real important. So it has to catch their imagination, because it's a lot of work for them, and they have to be able to maintain that excitement about what they're doing.
Monica, the flood of public response to the Melatonin is interesting. Both Time magazine, even the science journals, predicting great waves of possibilities.
Yeah.
It was generated a kind of a counter, an undertone, or the reaction that maybe it's being overstated. Between those two, what is your position? Are you optimistic about research in melatonin and its benefits to?
I think it's probably overstated at this point. One of the concerns with melatonin is that you could buy it over a counter. It's not a prescription drug at all. So you could go to a health food store and buy it and take it in mega doses. And as a matter of fact, they can't kill a rat with it. I mean, you can't give a rat enough of it to kill it. So it's really pretty safe. So there's, and I think that's part of the undertow, is that people would maybe tend to experiment themselves with it. But whenever something like this comes out, it always causes a lot more commotion. And then no one drug is going to be anti-aging. I say this, and tomorrow we'll come out with one. Or completely have an effect on the immune system. It's always more complicated than that. But the drug has a lot of potential. It definitely seems to work for sleep. If you jet lag, So and the fact that older people, people in their 80s, don't produce very much of it at all, generally. Whereas young people produce high doses, that seems to speak for that it might have something to do with the aging process. Yeah, I think it has potential, but it's probably not going to be as great as it sounds right now. I haven't bought any and ingested it myself.
Let us know when you do.
Yeah. There's a difference.
Monica, why are people at DuPont interested in studying photosynthesis?
DuPont is very interested in all aspects of photosynthesis because they make so many pesticides and drugs that affect plants. And they really would like to figure out how photosynthesis works, the mechanism, so that they could improve plants. And they would like to genetically engineer plants that could withstand certain pesticides, that could withstand antifungal agents, things like that. So they're very interested in photosynthesis. And quite frankly, if you could figure out photosynthesis and make it more efficient, there's a lot of money in that. You could genetically engineer things to be more efficient. We talked about photorespiration. If they could get rid of the enzymes aren't always as efficient, they might react with the oxygen instead of the CO2. So they're very interested in it. The person that figures out, I said this tunnel vision at the Gordon conference where they're trying to figure out how to split water. To me, I don't care about the manganese, and it made me think that my students might not care about some things too. But the person who actually figures that out will probably win the Nobel Prize. The person who figures out how chemically water is split in the process of photosynthesis. Because if we could split water, we could make we could make oxygen. We could split water and make more oxygen. There might be a technique that we could use for different things. I can't think of anything right now where we want to use it, I'm sure.
Space travel.
What?
Space travel, man.
Oh, all right. Yeah, but why would you want to carry water? It would be more efficient just to carry oxygen. Water's heavy. Yeah.
Some biologists have reacted very negatively to the Human Genome Project, claiming that, say, for example, that I'm not sure about the percentage, but say only 10% of human diseases are genetic in origin, and yet we're investing a great deal of money into mapping the genome. And so some biologists have found that's kind of a waste of the money that we're putting into that. What's your reaction?
I think it's one of those things that you can't turn back. It probably does take a lot of money to sequence the human genome, but it's going to happen because there's just too much potential information that can be gotten. And it's not just the human genome they're sequencing. They're sequencing everything. Every organism out there they're trying to get an entire sequence for. I think right now there's one bacterium that they've got that they know everything. They know all the sequences. And the amount of information you can get from that is tremendous. And maybe not diseases. Maybe you won't figure out all the diseases, but the control mechanisms. If you could just make comparisons between different organisms, how things are turned on, how they're turned off, what the timing is. I mean, there are a lot of things where the timing is just wrong, or it's being turned on in the wrong cell. Or what if you could turn on the gene that just makes nerve cells grow? Can you kind of make the nerve cells regenerate? Well, that would take care of paraplegics, things like that. So the information is somewhere there in the genome. All the information is there. And that's what I mean. I always felt that once you got all this information, you knew everything. But you just knew how to control things. You really didn't. I hope you understand that. You really won't know what's going to happen because it's all interactive. It's all connected. So you change one thing and that changes something else. And you don't know enough about everything. There's no way that you could control it. So we might actually speed up evolution, but we're not going to control evolution.
In the area of complex adaptive systems, in the studies as they exist right now, is there one sub-area, economics, biology, physics,
an awful lot of work with complex systems, really with the DNA. See, I sort of zero in on that because that's what I'm familiar with. So they're actually looking at the DNA as a complex system, and they're saying, are there sequences or is there something in there that we're not seeing? They're working with algorithms, genetic algorithms, trying to determine what's the shortest thing that you have to specify this particular organism. But as far as figuring out these universal laws for complex systems, I don't know if they're close there. And some scientists that I met when I visited there didn't think that we would ever find any universal laws for complex systems. But just studying them, or just having that viewpoint about life or about things, would give you a different perspective as to how you set up your experiments. So that you would be looking at them different. They're doing a lot with the economy. They've done a lot with weather. And you wouldn't think offhand that weather should be related to evolution. But when you think about it, the way they present it, there are similarities. So I don't know where they're going to break through. And there are certain graduate schools that are working on this. There are no undergraduate institutions that have really tried to tackle this because it is complex. But I think it kind of follows from the kinds of things, well, in my mind, it kind of follows what I've been doing, and I think it's pretty exciting. So it's a direction I would like to go.
There was research you said didn't work because of that certain piece of equipment. Did they have that equipment at DuPont?
Yeah, they do. And we have it now, I might add. We have the sonicator now, so I could go back and redo those experiments. And a sonicator is just something that vibrates. So it makes a real high noise, and it breaks open the cells without really hurting the enzymes or anything. I suppose if you did it long enough or hard enough, eventually it could disrupt all kinds of things. But it just breaks open the cells, and things like bacterial and yeast cells are very hard to break open. You would think they wouldn't be, but they are. So then once you start using chemicals to break them open, you're changing, you're having an effect on what you're measuring, plus it just isn't as accurate. Yeah, so we have a sonicator now. Anybody wants to use one for any reason. It's good for cleaning diamond rings and things too. I don't have any diamond rings. Just back there. Any other questions?
Well, Monica, thank you. Thank you very much. Also, let me announce before you go that outside here is a refreshment area, and you'll have a chance to talk to Dr. Becker and get some refreshments. So thank you all for being here.